Renal mechanism for pH regulation. The mechanism of regulation of blood pH involves the following factors. A. Frontline defense. There are mainly buffer systems in the blood, which restricts pH changes in the blood body fluids. Second one, respiratory mechanism. Regulation of excretion of carbon dioxide and hence regulation of carbonic acid concentration in extracellular fluid. B. Second line defense. This is achieved by kidneys. Ultimately, excretion of excess of acid or base, and thus ultimate regulation of concentration of hydrogen and bicarbonate ions in extracellular fluid. C. Dilution factor. The acid introduced into and formed in the body are distributed throughout the extracellular fluid. Although this may not properly be regarded as a regulatory mechanism, entrance of a given amount of acid into a smaller volume of fluid, as in conditions of severe dehydration, results in relatively greater rise in hydrogen ion concentration and decrease in effective buffer base. The normal pH of blood is maintained in the narrow range of 7.35 to 7.45. That is slightly alkaline. Production of acids by the body. The metabolism of the body is accompanied by an overall production of acids. These include the volatile acids like carbonic acid or non-volatile acids such as lactic acid, sulfuric acid, or phosphoric acid. Carbonic acid is formed from the metabolic products of carbon dioxide. Lactic acid is produced in anaerobic metabolism. Sulfuric acid is generated from proteins, especially the sulfur-containing amino acids. Phosphoric acid is derived from organic phosphates, example, phospholipids. All these acids add up the hydrogen ions to the blood. Production of bases by the body. The formation of basic compounds in the body in the normal circumstances is negligible. Some amount of bicarbonate is generated from the organic acids such as lactate and citrate. A vegetarian diet has a tendency for a net production of bases. This is due to the fact that vegetarian diet produces salts of organic acids such as sodium lactate, which can utilize hydrogen ions produced in the body. For this reason, a vegetarian diet has an alkalizing effect on the body. The body has developed 3 lines of defense to regulate the body's acid-base balance and maintain the blood pH around the normal blood pH 7.4. That is blood buffers, respiratory mechanism, and renal mechanism. We will be concentrating on renal mechanism. Kidneys affect the acid-base equilibrium by providing for elimination of non-volatile acids, already explained as lactic acid, carbonic acid, ketone bodies, etc. After being buffered with cations, principally the sodium ions, are first removed by glomerular filtration. Body cannot afford to lose sodium, being extremely important. It is recovered in the renal tubules by reabsorption in exchange of hydrogen ions, which are secreted. It is recovered as sodium bicarbonate, also known as the alkali reserve. The renal mechanism of pH regulation. The renal mechanism tries to improve or provide a permanent solution to acid-base disturbances. This is in contrast to temporary buffering system and a short-term respiratory mechanism. Kidneys regulate the blood pH by maintaining the alkali reserve. Besides excreting or reabsorbing the acid or basic substances as the situation demands. Urine pH normally lowers the blood pH. The pH of urine is normally acidic, approximately 6. This clearly indicates that the kidneys have contributed to the acidification of urine when it is formed from the blood plasma of pH 7.4. In other words, the hydrogen ions generated in the body in the normal circumstances are eliminated by acidified urine. Hence, the pH of urine is normally acidic, approximately 6, while that of blood is alkaline, that is 7.4 pH. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase is of central importance in the renal regulation of pH, which occurs by the following mechanisms, like the excretion of hydrogen ions, second one, reabsorption of bicarbonate, third one, excretion of titratable acids, the fourth one, excretion of ammonium ions. Carbonic anhydrase. It is a zinc-containing metal enzyme. It especially catalyzes the removal of carbon dioxide from carbonic acid. At the tissues, the formation of carbonic acid from carbon dioxide and water is also accelerated by carbonic anhydrase. The enzyme is present in red blood cells where it associates with hemoglobin, but never found in plasma. In most of the tissues, where catalyzes formation of carbonic acid from water and metabolic carbon dioxide, and in parietal cells of stomach, where the enzyme is involved in secretion of hydrochloric acid, in renal tubular epithelial cells. And recently, it has been demonstrated in small quantities in muscle tissues, pancreas, and even in spermatozoa. First, we'll see the excretion of hydrogen ions. Kidney is the only route through which the hydrogen ions can be eliminated from the body. The hydrogen ions excretion occurs in the proximal convoluted tubule or the renal tubular cells and is coupled with regeneration of bicarbonate. Carbonic anhydrase is the chief enzyme that catalyzes the production of carbonic acid from carbon dioxide and water in the renal tubular cells. Carbonic acid then dissociates to hydrogen ions and bicarbonate. The hydrogen ions are secreted into the tubular lumen in exchange of sodium. The sodium in association with bicarbonate is reabsorbed into the blood. This is an effective mechanism to eliminate acids or H+ ions from the body with a simultaneous generation of bicarbonate. The latter adds up to alkaline reserve of the body. The hydrogen combines with a non-carbonate base and is excreted in the urine. Second one, reabsorption of bicarbonate. This mechanism is primarily responsible to conserve the blood bicarbonate with a simultaneous excretion of hydrogen ions. The normal urine is almost free from bicarbonate. This is explained as follows. Bicarbonate freely diffuse from the plasma into the tubular lumen. Here, the bicarbonate combines with hydrogen, secreted by the tubular cells to form carbonic acid. The carbonic acid is then cleaved by carbonic anhydrase, the enzyme, of tubular cell membrane to form carbon dioxide and water. As the concentration of carbon dioxide builds up in the lumen, it diffuses into the tubular cells along with the concentration of gradient. In the tubular cell, carbon dioxide again combines with water to form bicarbonate, which then dissociates into hydrogen ion and the bicarbonate. The H plus ions or hydrogen ions secreted into the lumen is exchanged for sodium. The bicarbonate is reabsorbed into the plasma in association with sodium. Reabsorption of bicarbonate is a cyclic process with the net exclusion of H plus ions or generation of new bicarbonate. This is because the H plus is derived from the water molecules. Third one, excretion of titratable acid. Titratable acid is a measure of acid excreted into the urine by the kidney. This can be estimated by titrating urine back to the normal pH of blood, around 7.4. In quantitative terms, titratable acidity refers to the number of milliliters of N/10 sodium hydroxide rocaditrate 1 liter of urine to pH 7.4. Titratable acidity reflects the H plus ions excreted into the urine, which results in a fall of pH from 7.4 that of blood. The excretion of H plus ions are actually buffered in the urine by a phosphate buffer. H plus ions is excreted into the tubular lumen in exchange of sodium ion. This sodium is obtained from the base disodium hydrogen phosphate. The latter, in turn, combines with the H plus to produce the acid sodium dihydrogen phosphate, in which form the major quantity of titratable acid in urine is present. As the tubular fluid moves down the renal tubules, more and more hydrogen ions are added, resulting in acidification of urine. This causes a fall in pH of urine to as low as 4.5. You can see this thing in this schematic representation. Fourth one, excretion of ammonium ions. This is another mechanism to buffer H plus ions secreted into the tubular fluid. The hydrogen ions combines with ammonia to form ammonium ion. The renal tubular cells deaminate glutamine to glutamate and ammonia. This reaction is catalyzed by enzyme glutaminase. The ammonia liberated in this reaction diffuses into the tubular lumen, where it combines with H plus ions to form ammonium ion. Ammonium ions cannot diffuse back into the tubular cells and therefore excreted into the urine. The summary of the renal regulation. The renal regulation of pH occurs by following mechanisms. First one, excretion of H plus ions. Second one, reabsorption of bicarbonate ions. Excretion of titratable acids. And fourth one, excretion of ammonium ions. Action of carbonic anhydrase inhibitor. The enzyme carbonic anhydrase, which catalyzes the important reaction in renal tubules by which the hydrogen ions are produced for secretion, may be inhibited by sulfonamide derivatives. The most potent inhibitor found in acetazolamide. When the drug is administered, the urine becomes alkaline. Increased amount of sodium bicarbonate appears in the urine. There is also reduction in titratable acidity and in ammonia excretion in the urine, with an increase in potassium excretion from distal tubular epithelial cells. All of the above can be explained by inhibiting the action of carbonic anhydrase due to induced loss of due to less hydrogen sodium exchange. Drug is usually clinically used as a diuretic to induce the loss of sodium and water in patients who will be benefited by such regime. Example, cognitive cardiac failure and in hypertensive heart diseases. Under normal conditions, the rate of hydrogen secretion is about 3.50 millimoles per minute, and the rate of filtration of bicarbonate ion is about 3.49 millimoles per minute. Hence, all the bicarbonate ions are normally reabsorbed, while a slight excess of hydrogen ions remain in the tubules to react with other substances and to be excreted in urine. Acid-base imbalance can manifest as acidosis and alkalosis. Acidosis, which can be either metabolic acidosis or respiratory acidosis, and alkalosis. This also can be metabolic alkalosis and respiratory alkalosis. Metabolic acidosis, it is also called as primary alkali deficit. It is the commonest disturbance of acid-base balance observed clinically. It is caused when there is a reduction in the plasma bicarbonate with either no or little change in the carbonic acid fraction. Respiratory acidosis. It is also called as primary carbonic acid excess. The underlying abnormality here is increase in carbonic acid in the blood, which follows decreased elimination of carbon dioxide in the pulmonary alveoli. Metabolic alkalosis is also called as primary alkali excess. This condition results from an absolute or relative increase in bicarbonate. Primary alkali excess or increase in the alkali reserve is the most frequent cause of clinically observed alkalosis. Respiratory alkalosis is also called as primary carbonic acid deficit. This condition occurs when there is a decrease in carbonic acid fraction with no corresponding change in bicarbonate in plasma. Excess quantities of carbon dioxide may be washed out of the blood by hyperventilation.